covering some important concepts in computational metagenomics, COMEG. So, what your data looks like after data to ASV table. So, you end up with an amplicon ASV, amplicon sequence variant table. Rows or columns are ASVs, exact biological sequences that passed data to denoising. And the other axis is samples. Each cell is the count of that ASV sample in that sample, and this is the high resolution replacement for 97% OTUs. OTUs and can resolve to species strain depending on marker gene variability. So you might right. So after denoising with data 2, you obtain the amplicon sequence variant ASV table. Rows are ASVs that are unique error corrected sequences, often resolved down to the strain level, and columns are samples and entries are the counts. This replaces older OTU tables which have 97% clustering. ASVs are more reproducible across studies because they are defined by exact sequences, not arbitrary cutoffs. The ASV table is the primary input for all downstream ecological and statistical analysis. Why normalization is needed? So raw ASV tables are dominated by sequencing depth effects. Some samples may have 50,000 reads, others 500,000. Without adjustment, higher depth samples will always appear richer regardless of biology. Therefore, before comparing samples, we must normalize to make samples comparable, right? And normalization ensures that patterns reflect biology, not just sequencing effort or technical noise. Okay. Now, normalization approaches we have. So, two strategies are presented. I'll take you over these strategies. So, firstly, we have relative abundance or TSS, which is total sum scaling. So, what it does is it divides counts in each sample by the total reads of that sample. So, relative abundance upon the total sum scaling. Divide counts in each sample by the total reads of that sample. Each sample becomes a vector of proportion summing to 1. So it is simple and interpretable, but introduces compositional constraints. Relative data can create spurious correlations. Then the next one we have is CLR, which is centered log ratio transform. So we take the log of each count divided by the geometric mean, GM, of all counts in the sample. This places data in Euclidean space, aligning with compositional data theory, and requires handling zeros, pseudo counts. So the choice matters. Blake, Curtis, or Jacquard often used relative abundances, TSS, this one, but Atchison distance requires CLR, centered log ratio. Now, alpha diversity concept. So alpha diversity is within sample diversity. So alpha diversity would be a metric which measures how many taxa are present, that is the richness, and how evenly they are distributed, that is their evenness. So it gives us a measure about the richness as well as the evenness of a particular taxa, right? Richness alone, just the number of species, misses balance. Evenness tells whether the community is dominated by a few taxa or balanced. And then we have alpha diversity indices which combine these concepts differently. Then we have the Shannon index. So the Shannon diversity H is equal to minus summation P log P, where Pi is the relative abundance of taxon I, right? So this is the Shannon index formula, and it captures both the richness as well as the evenness. So more the taxa, the higher the H value, and more equal abundances, the higher the Shannon index value. Sensitive, so it is sensitive to both common and rare taxa, interpreted as the information entropy of a community, implemented in R via vegan, these double colons, then diversity index is equal to Shannon, right? Moving on, Pillow evenness, so it is defined as J. Let me show you the formula. J is equal to H, which is the entropy, the Shannon index upon log S. H is actually the entropy only, because the formula is minus summation Pi log Pi, right? So the uncertainty, or you could say the entropy only, right? So this is the formula for that, and that denotes the Shannon index itself. And this is the formula for Pillow evenness. So it normalizes Shannon by maximum possible diversity. Log S is the maximum possible diversity, log of the species count, right? Provides a pure measure of evenness. J is equal to 1 means all taxa are equally abundant, and J less than 1 means that we have uneven distribution, that there is some dominance in some species while some species are subordinates or not dominant enough. And this is useful when we want to separate balance from richness effects. Now moving on, we'll be discussing about Simpson index, right? So the formula variance exists in general. It will measure the probability that two randomly selected individuals belong to the same species. Now it is more weighted towards abundant taxa, less sensitive to the rare tail compared to Shannon. Now low Simpson index means that it is diverse, that is there is low dominance, and high Simpson index means dominated by a few taxa, right? And implemented in R via vegan double colon diversity index is equal to Simpson. In practice, we use use Simpson alongside Shannon to detect whether differences are in the common core or the rare biosphere, right? So measures probability that two randomly selected individuals belong to the same species, and it says less sensitive to the rare tail compared to Shannon. Now rarefaction curves, another important concept. So rarefaction plots species richness against the number of reads sampled or individual subsampled. The purpose of rarefaction curves is to check if sequencing captured most diversity or if more depth would reveal more taxa. If curve plateaus, sample is sufficient. If it keeps rising, you are still undersampling. In practice, rarefaction is visualized via vegan double colon rare curve. Biological insight is a shallow plateau may indicate a simple environment, for example, vaginal microbiome, while a steep rising curve suggests a hyperdiverse system, for example, soil. Now, why rarefaction matters? So it ensures that we can trust diversity comparisons. If one sample's curve plateaus and another's keeps rising, their apparent richness difference may simply be an artifact of sequencing depth. Rarefaction thus validates alpha diversity metrics and prevents misinterpretation. Now, moving on, we are going to be discussing beta diversity. So that is between sample differences, right? So beta diversity measures how communities differ from each other. The workflow is how communities differ from each other. Workflow is start with the species or the ASV cross sample matrix, then choose a distance or dissimilarity metric, then build a sample cross sample distance matrix, and then use ordination. That is, for example, principal coordinate analysis PCOA. This is not principal component analysis. This is principal coordinate analysis. So PCOA to visualize, and this is the backbone of comparative microbiome studies to measure how communities differ from each other. Now, distance measures, some families I'm discussing. So, detection based is Jacquard, that is checking the presence or absence. Then, abundance based is Bray-Curtis and Euclidean. Compositional is Hesson, that is CLR plus Euclidean. Then, rank based, Kendall, Spearman, one minus correlation. Then, phylogenetic, Unifrac, which is unweighted presence or absence, and weighted is abundance. Each captures a different ecological lens. Now, Jacquard distance, so is Jacquard is equal to 1 minus the intersection set, the number of elements in the intersection set. Let me write it down clearly. So Jacquard formula is 1 minus A intersection B, right? So the number, this is the mod that we have applied. So the number of elements in or species in intersection upon the number of elements in or species in the union, right? So it compares sets of taxa, ignores abundance, and this is the formula for Jacquard index distance. High Jacquard means that the two samples share few taxa. Why would this be? Because see, this is the value that has been given to us, and let me show you via Venn diagram. So A and B, this is right. So you can see that the intersection is very small here. So that means the numerator is very small, and the denominator, that is A intersection, sorry, A union B, is very large. So this value actually becomes a small value. 1 minus small would give us a higher J value, right? Close to 1, right? It is close to 1, let us say. This is the first example. Then second example is that we have another Venn diagram here, that is A, and you see B as well. So you see the intersection is considerably higher than this case. So this is J1, and J2 would be 1 minus A intersection B. Now A intersection B is actually greater than the first case, right? And A union B would be the same, right? So what this does is that this value has actually increased. This value has increased. So 1 minus a greater value. So a greater value than the one in the first case would be meaning that we have an overall smaller than value, smaller than the previous case. So that means that high Jacquard, that is in this case, two samples share few taxa. So now it makes sense that the intersection is very less. That is, they are sharing very few taxa, A and B. So that is why it is yielding a very high J value. In case we have more intersection, that is more taxa are shared, there is more intersection between the species A and B. That means that we will have an overall smaller Jacquard value. So that is the relation that I've explained. So it is very logical, right? You just need to remember the formula, and then you can derive all these results on your own as well. Now, useful when presence or absence is key. That is, for example, pathogen surveillance. And in R, we implement is using a wedge dist method is equal to Jacquard, and it is useful when presence or absence is key, right? So all of these points you must take into consideration. Then we have the Bray-Curtis distance. So formula is 1 minus 2 times shared abundance, 1 minus 2 times shared abundance, and upon total abundance, right? Total. This is the formula for Bray-Curtis distance. It is sensitive to abundance profiles, downweights joint absences, widely used in microbiome ecology because it reflects shared community structure. Now, higher values means more dissimilar in abundances, and we use the method Bray. Then, Hesson distance handles compositional nature of relative abundance data. Workflow is apply CLR transform, log ratio of each taxon to geometric mean, the one we discussed, CLR. Then compute the Euclidean distance. Advantage is that it respects the fact that microbiome data live in a simplex, that is a relative scale, implemented with R's compositions package, right? Moving on, we have rank-based distances now, Kendall and Spearman. So distance is equal to 1 minus correlation between abundance ranks of taxa across samples. Now it focuses on reordering of community structure. If dominant taxa stay dominant, distance is low, even if rare taxa differ. If rankings shuffle, distance is high. It is useful for detecting ecological restructuring versus mere shifts in rare taxa. Now, comparing distances, so detection or abundance, Jacquard, Bray-Curtis, Hesson, are driven by which taxa are shared or absent, especially rarer taxa. Now, we have rank-based distances highlight changes in dominant structure who is top versus bottom. Now, thus, choice of metric changes biological interpretation. Then we have unifrac unweighted, which incorporates the phylogenetic tree of taxa, and the distance is the fraction of total branch length unique to one sample or the other. Captures phylogenetic turnover. Communities may look similar by taxonomy, but differ in deep evolutionary lineages. So this is the phylogenetic turnover, that communities looking similar in the taxonomy, but when we dive deep, we see that they differ in their evolutionary lineages. Especially useful when taxonomic labels are coarse or incomplete. Now, unifrac weighted, same tree as unweighted, but weights by abundance differences along each branch. So it captures abundance weighted phylogenetic shifts. Two samples with same lineages but very different abundances will show distance. Offers a richer, abundance sensitive view of phylogenetic community structure. Now moving on, we have GUnifrac R package. So practical note, our GUnifrac package implements both the weighted as well as the unweighted unifrac. We give the input as OTU or ASV table plus the phylogenetic tree. The output that comes is unifrac distance matrices for downstream ordination, PCOA, that is principal coordinate analysis, and NMDS.